Alright guys, in today's lesson we're going to talk about proteins, and so as I go through this lesson I expect you guys to take notes on the proteins. This is going to go through a couple animations. I'm going to explain how enzymes work. I'm going to explain what proteins are, and then the sub's going to pass out a worksheet for you guys to work on in class. I expect it to be individual work, and you'll hold on to it until Monday, and you have no homework over the weekend. Yes, fourth period, you have no homework over the weekend, so hopefully that makes you guys happy. Alright, so far we've talked about carbohydrates and we've talked about lipids, which are both macromolecules, and proteins is a third type of macromolecule that we're talking about. And proteins do all kinds of things for us. Proteins are the basic building block of our bodies. They perform all kinds of functions in the cell. But most of them are structural proteins, and most of them help speed up chemical reactions. So enzymes is an example of a protein. Proteins also serve as transport carriers, basically things that help move large materials from one area to another. They act as antibodies, and they allow materials to go across our cell membranes, which are made of lipids. I'll give you guys a couple minutes here to take down some of these different functions of a protein. My suggestion would be to just make bullet points. Make bullet points for these four things. Alright, and if at any point you need to go back or you need to get notes, just take them from a neighbor that's sitting next to you when this presentation is over. Alright, let's go ahead and look at enzymes here. And this is going to pull up an animation for us.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme called...
And I'm just going to turn the audio off and I'm going to talk my way through it. Basically, let me back this up a bit. Basically what we're looking at here is an enzyme, and I showed you guys this animation before, but before you guys were introduced to an enzyme. So let's just first, real quickly, enzymes are proteins, okay? And one of the functions of proteins is to help facilitate chemical reactions. So this purple blob right here is an enzyme, and these cutouts that you see are a part of the enzyme that's called the active site. So as this enzyme, and you can see it's labeled right there, active site. So this enzyme is found somewhere in our body, and they're all over our body, and they take part in all kinds of chemical reactions. And these two things right here that are going to come into the active site is a generic term called substrates. Okay? And substrates could be any number of things, but most typically what they are is they are compounds that are joined together at the location of the enzyme to form larger compounds. Example would be these could be two monosaccharides, and at the enzyme they could join together to form a disaccharide. So we have these two substrates that are coming in, and these two substrates, when they're together, is called an enzyme substrate complex. And so what happens is when these things connect together, remember these are just molecules, so there are a whole bunch of different atoms and they're a three-dimensional shape, and when they come in contact with each other, they change shape. Okay? And they change shape because of a chemical reaction that happens when they bind to the enzyme. The enzyme acts as a catalyst. It speeds up chemical reactions. So these two substrates come in, they come into the enzyme, they change shape, and they form a new structure. And this new structure now has these two substrates joined together, and then the substrate is released, and what we end up with is a product. So this is a chemical reaction. So what's coming in are these substrates, and these substrates serve as our reactants. They join up with the enzyme changes their shape, and they're released as a product. So basically the enzyme is a location, actually more specifically the active site on an enzyme is the location at which a chemical reaction takes place. Okay, and that's just the basic idea about it. We call these things catalysts because a catalyst is something that speeds something else up. Okay, so let's go ahead back here and look at our slide. I'm going to go ahead and move to the next. This is showing you a couple of things, and I'm going to go ahead and make this interactive. And this is showing you guys a couple of things, and this must have been what Mr. Hopkins was talking about with the screw up. Okay. These pictures that you see here are amino acids. Okay, alanine is an amino acid, valine is an amino acid, cysteine is an amino acid. Notice that they all end in INE. So anytime you see a word that ends in INE, you can automatically think of an amino acid. Okay, so these amino acids make up proteins. So protein is the polymer for an amino acid. Okay, so a bunch of amino acids joined together is a protein. Okay, just like a bunch of monosaccharides joined together is a polysaccharide, a bunch of triglycerides bonded together is a fat or is a lipid. A bunch of amino acids joined together is a protein. And then what you see here is these are the structural groups of alanine. So you can see that they're pretty big and complex molecules, and if you notice, there's a functional group on each one of these, the COOH, which is the COO or the carboxyl group. There's the COO or carboxyl group. And these are the two ways that we could draw alanine, and these are the two ways we could draw valine. And then highlighted in blue is something called the R group. And the R group is something that helps define what the amino acid is. And we'll talk more about this in detail. But what I want you guys to take away from this is the fact that our amino acids contain carboxyl groups. And it makes sense because I told you carboxyl groups are found in lipids and proteins, and that proteins are simply polymers of amino acids, meaning that many amino acids joined together will form a protein. Alright, let's look at this next slide. The bonds that hold the amino acids together are called peptide bonds. So far we've talked about hydrogen bonds, ionic bonds, covalent bonds, and now here's another one called a peptide bond. Well, a peptide bond is simply a bond that holds two amino acids together. When we have two amino acids that are joined together, we call it a dipeptide. Sorry about that. We call it a dipeptide. So the dipeptide is two amino acids joined together by a peptide bond. Notice that in this chemical reaction that you see up here, we have two single units or two amino acids that are coming together, and as a result water is released. Because water is released, this is a condensation synthesis or a condensation reaction. If we were taking this chemical reaction in the opposite direction and we were using water and splitting it, it would be called hydrolysis or water splitting. Alright, how enzymes work. There is a short video that I'm going to ask the sub to play for you at the conclusion of this presentation, and when that time comes, my instructions have in there how to play that video and everything. So let's go to the next slide. Okay, what do enzymes do for us? Well, we know enzymes are proteins, and we know proteins are long chains of amino acids. Well, enzymes can help regulate our chemical pathways. Which means that they can make materials that the cells need, they can release energy, or they can transfer information. Enzymes also catalyze or speed up a type of chemical reaction. And that's because each enzyme will fit only a specific type of molecule. So by having this specific type of molecule that only fits a specific type of enzyme, our body can easily control the pathways at which it does things. So, for example, if a hormone is released, that hormone can only go into the appropriate receptor or the appropriate enzyme. And so it's a way that your body can basically communicate with specific parts without interrupting all of the other parts. So enzymes regulate chemical pathways and they speed up chemical reactions. Alright, so an enzyme substrate is the specific reactant that's going to react with it. So just like the animation that I showed you the picture of the two substrates that were going to connect to the enzyme. Well, they had these goofy looking shapes because they are specifically, I should say, the enzyme is specifically designed to accept them or to allow them to bind to the active site. So each particular substrate, like this one right here, let's back up a second. Like this one right here is intended to fit this active site on this enzyme. Okay. Let's just move to the next slide. So the fit that exists between the substrate and this enzyme is not a rigid fit. Okay, so as a substrate enters, the active site changes its shape slightly to fit the substrate really snug. And this gives a place for a chemical reaction to take place. Alright, so sometimes what this does is it can take a complex enzyme or a substrate and it can break it into two. Or sometimes it can take two separate substrates and join them into one. And the example that I showed you on the animation earlier was two different substrates that were being connected and attached to each other to make a product that was basically twice the size as the original substrates. This particular illustration is showing a substrate come into an enzyme. The enzyme will fit snugly. It'll break bonds that exist between two molecules of that substrate, and then the products end up separate from each other because that bond was broken. An enzyme has the ability to lower the activation energy, and it does this by accepting reactant molecules. Basically what I want you to take away from that is the fact that the energy needed to start a chemical reaction is called activation energy. An enzyme has the ability to lower that activation energy, which is really important for our body. It makes our body more efficient so we don't have to use as much energy to do the things that we do. Enzymes have the ability to form larger molecules from smaller molecules, or the very opposite. It can take smaller molecules and make larger molecules out of it. It's the enzyme size and shape that make it function the way that it does. So if its size changes or its shape changes, the enzyme itself won't be able to do the job it was intended to do. Alright, and this is the last slide I'm going to cover. I just said in the previous slide that enzyme shape and size are very important to what it does. Now, its shape is sensitive to its surroundings. So if there's a change in temperature, let's say that you get sick and you run a fever, many of the enzymes in your body are going to be denatured, which means that they're going to change shape because the temperature has changed. If the pH changes in an enzyme's environment, it'll denature as well. And we'll do a lab to explore this on Monday that shows exactly how enzymes denature, and you'll get to see it in a lab environment. So this is why cells can only survive and function within certain ranges of pH or certain ranges of temperature. Okay, at this time you can exit out of the movie and go back to the thumb drive and open up the video for the students to see. And once that's done, then we can start the worksheet.